Hello everyone, welcome back to my lessons. So today we will be doing exam questions for DNA Code of Life. So I am repeating this question because I noticed that on the video that the last part of this video was silent, and I'm really sorry about that. So the question said that study the diagram below, and let's look at the diagram. We have protein synthesis here. At A we have transcription, at B we have translation. At A we have a DNA molecule there, and then we have mRNA, which does not have the nitrogenous bases. It shows that it left the nucleus to the ribosome. Let's look at mRNA at 4. So we have mRNA which has the nitrogenous bases. These nitrogenous bases on mRNA, they pair with the ones on the DNA molecule at A. So if you look at G at the bottom, it pairs with C on the, starting from the left, on the mRNA. We have C on the DNA, we have G on the mRNA, we have G, we have C this side, we have T, we have A this side, because T pairs with A, T pairs with A, T pairs with A. The third base triplets on the DNA molecule, we have ACA. Let's look at this side, we have 6, 7, 8. Now let's look at the question. They say that name the process represented by A and B combined. A and B combined, A is transcription, B is translation. They are called protein synthesis. Where in the cell does process B occur? Process B occurs in the cytoplasm. Identify the following. Molecule 1. Molecule 1 is a DNA molecule. Polymer 3. Polymer 3 is a protein because we have the amino acids combined there. Give the correct base sequence to replace numbers 6, 7, 8. We talked about it. We have the first base triplets from the bottom on the DNA molecule, GCG, which pairs with CGC on the mRNA from the left. We have TTT, we have AAA, we have ACA, and then this side we have 6, 7, 8. Now, how do we find the 6, 7, 8 that they are looking for in 2.1.4? So we're going to write a sequence. Now look at this sequence. Without this sequence, you can never find the answers. Now let's look at this sequence. We have DNA, write DNA, and then write mRNA, and the mRNA write tRNA. You are given ACA, and from ACA they want mRNA, and then mRNA is under DNA. A pairs with U, C pairs with G, A pairs with U. If they wanted tRNA and you're given ACA, you move from DNA to mRNA first, and you move from mRNA to tRNA. You don't just move from DNA to tRNA straight. So you move from ACA to UGU, from UGU to ACA. As you can see, the DNA is the same as the tRNA, but you need to use this sequence. This sequence is very important. So let's look at the next question. We have write down the antigon label at label 2. So the first tRNA there pairs with CGC, the second one AAA, the third one 6, 7, 8. So it means that this one tRNA, it pairs with UGC. So if we have the mRNA UGC, the base triplets UGC, what is the base triplets for this tRNA? You use the same sequence again. You write DNA, under DNA you write mRNA, under mRNA you write tRNA, and then you're given UGC, which is mRNA. From mRNA to tRNA, you don't have to write the DNA. You just move. U pairs with A, G pairs with C, C pairs with G. So the answer is ACG. Now let's look at the next question. What is the DNA base triplet for glycine? Let's look at glycine. Glycine, we have tRNA this side, which is CCA. So we are looking for DNA base triplets. We are given tRNA base triplets, which are CCA. We use the same sequence again. We're given tRNA CCA. mRNA is, we need to move from tRNA to mRNA first. So the answer is GGU for mRNA. Now to DNA, the answer is going to be CCA. So number 1.2.1.6, the answer is CCA. Now let's look at the next question. Name the amino acid found at labeled 5. At labeled 5, we have tRNA. The base triplets for tRNA are unknown, but we know that it pairs with the base triplets on mRNA, which are AAA. So AAA, we're going to use the same sequence from mRNA to tRNA. You don't have to write the DNA. From AAA, A pairs with U, A pairs with U, A pairs with U in RNA. So the answer is UUU. So let's look at the tRNA on the table. We have UU there, and then UUU, the amino acid for UUU is lysine. The answer is lysine. Explain how the composition of a protein would change if the base sequence of the codon on molecule 4 from the left was UUU instead of AAA. So what would happen is that the antigon on tRNA would be AAA, then therefore amino acid would be phenylalanine instead of lysine. Thank you so much for watching. I hope you understand. Please don't forget to subscribe to the channel. I would really appreciate it. Thank you.